Hello dear students, welcome to Dr. JPL video lecture series. This lecture deals with Southern blotting falling under Unit 4A Immunoblotting Techniques as per Pharmacy Council of India syllabus for 3rd year B Pharmacy 6th semester, subject Pharmaceutical Biotechnology, subject code is BP605T. Myself, Dr. J.P. Lavande, Associate Professor, Department of Pharmaceutics, Shivlingeshwar College of Pharmacy, Almala, Taluka Ausa, District Latur. Dear students, we will going to study Southern blotting in different headings like Introduction to the Southern blotting, General Procedure for blotting, what is the Southern blotting, what is its principle, what is its procedure and applications involved in it. Just in order to make the content brief, in this video we will go through the introductory point related with the blotting techniques. Blotting is used in molecular biology for the identification of proteins and nucleic acids and is widely used for diagnostic purposes. This technique immobilizes the molecule of interest on a support, which is a nitrocellulose membrane or nylon. It uses hybridization techniques for the identification of the specific nucleic acids and genes. The blotting technique is a tool used in the identification of biomolecules such as DNA, messenger RNA, and protein during different stages of gene expression. Protein synthesis involves expression of a DNA segment which gets converted to mRNA to produce the respective protein. Molecules such as DNA, RNA, and proteins are subjected to biochemistry analysis, which are separated using blotting techniques. In the case of a cell, these molecules are present altogether, and hence with the help of blotting, scientists are able to recognize a specific molecule out of all others. Blotting is performed by allowing a mixture of molecules of interest pass through a block of gel which separates the molecules based on their molecular sizes. The hence processed molecules are required to be hard pressed against a suitable membrane, which will in turn transfer the molecules from the gel onto a suitable membrane, either nylon or nitrocellulose or PVDF, via capillary action. After the molecules are transferred to the membrane, their position does not change. Southern blotting was introduced by Edwin Southern in 1975 as a method to detect specific sequences of DNA and DNA samples. The other techniques of blotting emerged from this method have been termed as Northern blotting, which is used for RNA, Western blotting, which is used for proteins, Eastern blotting for post-translational protein modifications, and Southwestern for DNA-protein interaction blottings. Subtypes of blotting such as northern, western, and southern depends upon the target molecule that is being sought. When a DNA sequence is the foundation or code for a protein molecule, the particular DNA molecule of interest can be blotted using southern blotting technique. During gene expression, when the DNA is expressed as messenger RNA for a protein production, this process can be identified by northern blotting. Finally, the coded mRNA produces the concerned protein. This protein identification can be done by western blotting. Blotting approaches are viewed as an aid to the gel electrophoresis, which is generally applied for separation of DNA, RNA, protein, and yields reproducible results attributed to their excellent resolving power. Thereby, specific molecules can be detected amid the combination of molecules that are subjected to this separation. Most of the methods have a general step wherein the molecules of interest are transferred once separated are transferred from the gel to a solid membrane phase, which is accomplished by drenching in a solution across the gel and the membrane via penetrate paper. Many types of multifaceted apparatus are also provided from a lot of suppliers for electroblotting, which is more specifically useful for transfer initiated from less power polyacrylamide gels compared to commonly used porous agar gels. In case of DNA and RNA, the detection of specific sequences in the membrane are carried out via hybridization with nucleic acid labeled probes, which in the case of proteins is replaced by the use of labeled antibody probes. The initially developed protocol applied radioactive probes labeled with radioactive isotopes for detection purposes via implementation of autoradiography procedures. In this process, using the pattern of decay emission radiation radiated from a radioactive material is applied to produce an image on an X-ray film, which can also be made available as a digital image by application of scintillation based gas detectors or systems based on phosphorescence imaging. Keeping in mind the harmful effects of exposure to radioactivity, other kinds of labeling systems have been developed, which include fluorescent and chemiluminescent reagents. Luminescence, fluorescence-based methods are considered to be sensitive and background-free, thus generates more accurate results. Also, there are several other modifications that have been implemented in contrast to the original method, like DNA probes are more commonly applied rather than RNA. The nylon membranes have been replaced the traditional nitrocellulose to avoid the renaturation of DNA sequence during transfer. The transfer is carried out in alkaline, which was supposed to be in neutral solution as per the original protocol. The DNA is subjected to acid treatment for reducing its size in order to increase the transfer rate of larger fragments. The gel strips and tube gels applied in the original protocol are no longer used. Instead, gel formats are applied. Though there have been many changes in the original protocol, still the modern day protocol remains most of the fundamental features of the original protocol. Southern blotting was applied in many important studies like the genetic mapping of the human genome, which was based on blotting based detection of restriction fragment length polymorphisms. Also, the DNA fingerprinting was first developed via hybridization of the human DNA restriction digestion products with mini satellite probes. However, most of the primary applications of this method have been now substituted by DNA sequencing and polymerase chain reaction (PCR), as they provide more extensive facts or data and moreover are easy to implement. Nevertheless, blotting is still applied in a lot of arenas like in the measurement of copy number, analysis of long stretch of DNA that is difficult to be amplified or sequenced using PCR or DNA sequencing, and in case of structural analysis of DNA wherein the physical forms of DNA are separated using two-dimensional gel electrophoresis and thereafter it is detected using blotting of specific components. So in this video, dear students, we had gone through the brief introduction related with the blotting techniques. In the coming videos, we will go through general procedure for blotting, southern blotting, principle involved in southern blotting, its detailed procedure and its applications. So dear students, thank you for watching. If you like this video, please hit the like button, share our videos to your friends and subscribe to our YouTube channel Dr. JPL Video Lectures by pressing the red colored subscribe button to get notification about more such videos. Thank you, stay safe, take care and happy e-learning.